Okay, ladies and gentlemen, friends, colleagues, guest speakers, welcome to an update on MND Research, Homegrown and Abroad. My name is Carl Sunderland, I'm the General Manager of MND New Zealand. I will be seeing, I suppose, today, so that's very important. There's also some other people from our organisation here today. Claire Riley, who's our MND Community and Research Advisor. You'll hear a bit more from her shortly. We have Tony Foster, who's our support team leader. And what else have we got? I know Linda. We got Linda Oliver and Sally King, who are our support team members for Auckland and Northland. We have Kate Golders, who's our fundraising manager, next to Tony. We also have Greg Horton, who's on our National Council. Greg. So please feel free to ask them any questions you have today. So a bit of housekeeping. The toilets, including accessible toilets, are just around the corner by the red wall there. If there is an emergency, basically please make your way out of the nearest exit, and we all meet up on St John's Road on the pathway away from the building. The plan of the day today is for about an hour. We'll hear our first three speakers, Claire, Kerry and Emma. Then we'll have a break for about half an hour for afternoon tea, and then we will hear from Chris, and the panel will also be taking questions for about half an hour. We should be finished at 4:30, 5 o'clock at the latest. So today's, I'm just going to talk for a little bit, not too long, Claire, I promise. So today's event is part of Awareness Week 2019. So this is our second Awareness Week in our history, and we developed a concept called Cup of Tea for MND last year, which is basically, it's a chance for people to get together, to celebrate and remember, to raise awareness for motor neuron disease in their workplace or in their community, in their schools, and also to fundraise as well for the work we do. And we, last year we had about 50 events happened around the country, and Kate, we've probably got quite a few happening at the moment, but it's not too late if you want to hold a cup of tea event this weekend. Awareness Week doesn't finish till Sunday. We are a very small organisation. We do a lot. We punch well above our weight, and it's a really, really exciting time for our organisation at the moment. You might have seen some media this week with our TV superstar Greg, that we have become the official charity partner of Emirates Team New Zealand for the 36th America's Cup. So we are extremely honoured to be the official charity. In this partnership will go a long way to raising awareness of this devastating disease and the vital work we do. So that wasn't the only announcement we had in the last few weeks. We also announced our inaugural foundation corporate sponsor, Fulton Hogan. And what Fulton Hogan are a very practical company. And we identified through a community survey last year that there were some significant needs for practical support, and especially in the home for our community. So Fulton Hogan have come on with a 3-year partnership that will enable us to provide additional assistance to people living with motor neuron disease through the establishment of a home care fund for clients and for families. The MND New Zealand Support Fund, as we call it, will assist with practical support and our support team home visits. And we have to launch this very soon, probably next month. So that's an amazing step that's meeting an additional need that's been identified by our community. So thank you, Fulton Hogan. We've also been developing our new strategic plan, which will launch on the 1st of July for the next 3 years, to talk about our direction and where we want to go. And again, a lot of that is based on the survey that we did, which had an amazing response from our community last year. We've really listened to what people said they need and what we could do better, and the strategic plan will reflect that. So, also, don't forget, there's a little reminder, okay, that ignore that because that's last year's posting. So, our annual Walk to Defeat is happening again this year. We just hope to make it bigger and better every year. It's on Sunday the 10th of November. So this is our 5th year for this event. We also still need a few volunteers to help organise the Auckland Walk if anyone is interested and feels inspired this afternoon, and Kate will be glad to talk to you more about that over afternoon tea. But obviously we want as many people to attend and to fundraise because it's our huge flagship event. So, as I said, we are a very small organisation. You know, we achieve a lot. We receive very little government funding, and we rely almost completely on the generosity of the New Zealand community to continue the work we do. So what we do is basically, as I say, quite a lot. We provide free personalised emotional, social and practical support to people living with motor neuron disease and their families and carers. We also support health professionals, you know, who coordinate the care of people living with motor neuron disease and their families. You know, we provide up-to-date and trusted information on motor neuron disease. At the end of the day, we are the only charity focused on motor neuron disease in New Zealand, so it's our responsibility to do this. We advocate for the needs of the people living with motor neuron disease and their families. We develop awareness, hence Awareness Week, and understanding of motor neuron disease, because it still flies, as most all of you in the room know, so much under the radar. And we collaborate with others. We can't do this on our own. We need to work with other people, other organisations, with the health professionals, with our community, with our volunteers, so we can do this together. And of course, why we're here this afternoon is we also encourage and support research. So, as I say, that's what we're here to focus on. The last point this afternoon, to talk about research in New Zealand, actually talk about research developments and research within our organisation as well, and in New Zealand and overseas. So, without further ado, our first speaker is Claire Riley. So Claire is our MND Community and Research Advisor. Now, Claire's been part of the MND New Zealand team for several years, quite a while now, part of Dementia, in a good way. And Claire was instrumental in launching and coordinating our annual Walk to Defeat MND, our national community and fundraising flagship event, which is now our 5th year. During the first 4 years, the Walk to Defeat MND event raised almost $400,000 for support and research. Late last year, Claire took on a new role, as I say, as the MND Community and Research Advisor. She's working on a number of projects, including the collaborative effort in developing our inaugural research strategy, which we officially launched today. It is on the website if you want to read it, if you've got a bit of time reading on a Friday night. And Claire will tell you more about it now.
Thank you. Thank you, Carol. I wanted to take the opportunity today to officially launch our MND Research strategy for the next 3 years. The strategy is the result of a lot of work by a lot of people, of young team effort, and many of those people are here today, and I want to thank them personally. Just go to first of all, I want to go back to where this all began. As Carl said, our walks have become our main fundraiser, and in the last 4 years, with 50% of the funds going to research, we raised $400,000. Now, that might not seem a lot by international standards, but for a little organisation, it's not bad. And I think it's only fitting that I tell you what all your hard work has done and what we're doing with that money. So, going back a couple of years, the first 2 steps were to set up a registry, which was from a lot of work by Kerry, who's on the audience, and Dr. Richard Roxburgh and Miriam Rodriguez, and the importance of a registry so we know who people are with MND, where they're living, what type of disease they have. If we don't have that information, we can't connect them to the research that's happening. So that was a good first step. And then Dr. Scott, Emma Scott set up the research network, again fundamental, so that researchers can connect with each other and collaborate and know what each other is doing in terms of material research. So that was forming the foundation for our research program. Last year, some of you may recognise the community survey, and we stuck in a few questions about research because we wanted to know what was important to people with MND and their family in terms of what sort of research do you want us to do. Most people thought developing standard clinical pathways was important, followed closely by improving quality of life, finding a cure, and connecting New Zealand to global research. So I'll come back to the clinical pathways, but you can see that there are two things standing out. That's improving care and looking for cure. Yes, please.
So the aim of the MND New Zealand research strategy is to promote and foster a national MND research program to ensure all people with MND living in New Zealand receive the best available evidence-based care and support while remaining part of the international effort to identify the causes and ultimately a cure for MND.
So that's our overall vision, what we're working towards in terms of research. Now we found that our goals largely fell into 4 different areas. So I've mentioned cure and care, but the work we do more directly is communicate and also collaborate. And I'll talk a bit more about all of this in the next couple of slides. So here's what we're doing at the moment in terms of cure and care. Largely we're funding body, and as I said, we've been funding the Registry of the Research Network, and that will continue. Along with the others, we're helping to fund MND study, which is very exciting. I mentioned cervical pathways. This is quite a big project, and we decided the best thing to do was if we could get all the doctors around New Zealand who are fundamental in delivering care, all the specialists into one room, and we can have a meeting early to try and work out how we can go forward with national guidelines for material and care. So that will be happening in Wellington in September. I think of it in Wellington, the Minister of Health couldn't say no to come. So it's been held at Parliament, and the Minister of Health will be there. It's also important that we advocate for issues related to clinical care. And so last month we made a submission to the Health and Disability Review highlighting issues of importance to people with MND. And finally, next bit of exciting news is our first ever MND Research conference to be held here in Auckland in December. I'm sure this won't mind me saying that these are some of the real rock stars in motor neuron research throughout the world. So they're all coming here in Auckland in December, and tonight I've will be mostly directed at professionals and researchers, but it will be open to everyone, and we're all welcome. There'll be more details about that in the coming months. I'll also have a Ask the Experts session at the end, so you can ask all more questions. The other bit of the 4 C's is communicating globally, and this is basically about making sure we share all of our research information with you in a timely manner and in a way you can understand. So we want to have more of these face-to-face events where you can meet the researchers and ask questions. As Carl alluded to, it's also important we collaborate with bigger organisations such as the Neuron Foundation and Brain Research in New Zealand. So between Anna and ourselves, we're becoming a bit of a squeaky wheel trying to get on the agendas. So I think that's probably enough for this year. But guys, going forward, if we've got any researchers and the world of ideas, please come and talk to me. We're going to be having research wells and a scholarship for PhD students. I also want to make sure we have regular clinical sessions at Alexandra main centres, so that's going forward. Carl's really put this up, but mine's got the correct date. Just thank you again. All of these projects are directly funded by the community, so I hope you can see that we're trying to give the biggest bang for your buck.
How does the walk to defeat work? Do you get the money for walking or what?
Okay.
Because he gets sponsored.
So a lot of people decide to set up fundraising pages online to be able to get sponsored to walk. A lot of people who take part, they then get their workplace involved or their colleagues, or they do other events in support to then raise extra funds. And a lot of on the day itself, so there's a lot of activities on the day, so sometimes there's raffles or auctions and things like that, so you can get something back as well.
It was in about 16 different places around New Zealand, so you can just pick your place and turn up.
We were in Monica last year, there wasn't one there.
No, I think there was one in Ricardo. You can start one.
We just
Sorry, I'm just going to grab the mic again. On that note, so all of the walks are actually volunteer-led. So Claire started them up and she identified some incredible people around the country that stepped up, and they're organising these massive events, raising huge amounts of money. And then we support them from head office, but they are all completely organised and led by volunteers. So it's like people from our community that have stepped up, and so that's where the locations are, where those people have come forward and actually said that they're prepared to take that on.
Thank you. Any other questions? And I'll hand over to Kerry Walker from our registry. Thanks.
Thanks, Claire. So just introducing our second speaker. Kerry Walker is the curator of the New Zealand MND Registry. Kerry has worked in many areas of scientific research, including molecular biological lab research, clinical trials for new medicines, and large-scale epidemiological studies. I hate that word. She has been involved in the registry from its inception and manages all aspects of the study, from ethics and regulatory requirements, patient recruitment and data management. And Kerry is a great part of the MND New Zealand team. Thank you.
Thank you. Hi, I'm Kerry. I run the registry, and I'd like to just talk about a little bit about why registry, why it's important, what we do, how we do it, and why should we all be supporting it. So as Claire talked about, the Walk to Defeat fundraising was a game changer for research, MND research in New Zealand, and 50% of those funds that were raised were pledged to research. And so MND New Zealand thought long and hard about how do we spend the money and how do we get the best bang for our buck, basically. And research is really expensive, and so one of the first things they identified as a good way to spend money was to establish a registry. So patient registries can collect all clinical data from patients with a particular condition in one place. They themselves can then attract research as, if for example you're a pharmaceutical company and you wanted to run some research in New Zealand, who would you go to? Would you try and approach a clinician or maybe MND New Zealand? But with a registry we can say, yes, we have this number of patients, they live in this area, they have this type of disease, they have these genetic test results that we can provide you anonymously, of course. So that gives access to participants for research. It also means that we can collaborate with international registries and compare how are we doing in New Zealand compared to other Western countries. So our registry framework is really kindly offered to us by the Australian Motor Neuron Disease Registry. So that means that we collect the same type of data that they do. So we can say, in New Zealand, this is what happens with a person when a person develops this type of disease compared to Australia, which is a really powerful thing. So it was first conceived, we first had a meeting in November 2016, and then we launched in May 2017, and as you can see, Claire was the first participant, and she was signed up by Dr. Roxburgh, who's the clinician at the head of the registry. We had Beth Watson, who was the president of MND New Zealand at the time, and really integral in getting it started, and Paul Tallman, who's an Australian neurologist who runs the Australian Motor Neuron Disease Registry, and so kindly donated their kind of database framework for us to use. So the registry is essentially a research study, and that allows us to collect data about people who have motor neuron disease once they consent to. So that means that we have to stick to very strict ethical guidelines, and that we're based within the Auckland hospital, which allows access to kind of patient information and things like that. So basically, we're looking for anybody who has motor neuron disease in New Zealand who is interested in knowing if they would be eligible for any research that is currently available. So people with MND are eligible to enrol. Also people who have a positive genetic test for a motor neuron disease causing gene but don't have MND, or people who have a genetic form of motor neuron disease in their family. All of these people, please join the registry. So then we get everybody to consent. They sign a consent form which explains that their health data will be recorded, and then I collect all sorts of clinical information about everybody, kind of things like what was their first symptom, how long it took for them to be diagnosed, what kind of health services they use, all sorts of things. And then this is all stored anonymously in the Australian database, which is stored in the cloud. So then the registry is run by a steering committee, and that's made up of some people from MND New Zealand, some clinicians, some scientists, and they kind of give advice on how the registry should be run. And then, so the idea is that if somebody is looking to do research in New Zealand in motor neuron disease and they're looking for participants, they approach the registry and say, look, we would like to do this. We say, yes, that's a valid thing to be doing, that's a great idea, and make sure that their research is all ethically approved, and then we would approach people in New Zealand on the registry who would fit their requirements. So in New Zealand at the moment, we think, although a definitive study has never been done, but we think there are approximately 350-ish people in New Zealand with motor neuron disease at any one time. Currently we have 163 people on the registry, and we have some more people, more people who have since passed away, who we've recorded, we still have all of their data as well. So 163 people, and as you can see, they live all over New Zealand. I've recorded, we have all sorts of data about these people, which I won't show you today, just that all over New Zealand. We've released a journal article in the International Scientific Literature so that we are searchable by anybody who wants to find us in academia and research, basically. So this is this at the end of last year. So already we have been able to enrol people into research studies that are happening in New Zealand. So there was a research study run out of the Rose Centre at Canterbury University, where a team were looking at whether you could improve people's swallow based on a skill training program. We have had a study where a company was developing a technology that used brainwaves from your thoughts to control a computer interface. There was a study looking at stigma felt by people who have MND. That was an online survey. And our biggest study so far, and the one we're really excited about, is Dr. Scott's genetic research, which is about to get underway, where she's looking for, I'm sure she'll tell you more about it, but where she's looking for 300 people in New Zealand who have MND and going to run genetic screening on all of these people. Upcoming, there are a few studies. One about looking at blood-brain barrier, and one about whether assessments wheelchair assessments can be done remotely rather than requiring the person in the wheelchair to travel to their hospital or their clinician. Also, I'm really excited that we've been approached by some pharmaceutical companies, and we're really hoping that that will turn into our first ever clinical trial in New Zealand available to New Zealanders. So watch this space. So a registry is an important research tool basically, and it has a place in all parts of research. So from the very beginning of preclinical development, things that Professor Shaw's going to talk about, to clinical trials, and right to the very end, once there are, and there will be, drugs, good drugs for motor neuron disease, we will need to follow them and see how they work over time. So a registry is a really important thing. So thank you very much. Please do get in touch if you would like to know anything more about the registry, or if you'd like to enrol. You can get some more information about the registry on the MND New Zealand website, or please just give me an email or a call. Thank you.
Thanks, Kerry. One of the things that our support team do is when they visit families, that they'll talk about the registry and explain it and try and encourage people to join to see the benefits. So our next speaker is Dr. Emma Scotter, who is the head of the Motor Neuron Disease Lab at the Centre for Brain Research at the University of Auckland. Her team, in collaboration with Professors Mike Draganoff and Sir Richard Ford, researches various aspects of motor neuron disease. Dr. Scotter and her team have published articles revealing the higher death rates from MND and other countries using mortality data for New Zealand, and the presence of C9orf72 and UBQLN2 mutations as causes of amyotrophic ALS in New Zealand. Dr. Scotter, in collaboration with Associate Professor Richard Roxburgh, recently commenced a study which will perform genetic screening of people living with motor neuron disease in New Zealand. In 2017, their team established the New Zealand Motor Neuron Disease Research Network with funding from MND New Zealand. Dr. Scotter.
Thanks, everyone. Is there a slide advance? Okay, thanks everyone. I'll try not to talk for too long. I think Claire did just a wonderful job of giving us a picture of everything that's happening on the motor neuron disease NZ side of things and the great work they're doing. So we'll give a reasonably brief update of what my lab has been doing, and then we can hear from Chris how that connects to the international research. So before I do, I don't want to belabor the point, but I'll give you a quick rundown of how my team, who actually are here in the kind of fourth row, is most of the Scotter lab, so you can give us away. This is how we think about motor neuron disease and how motor neurons die in the disease. As you all will know, it's a progressive neurodegenerative disorder, and we're really interested to know whether the kind of 1 to 2 people affected per 100,000 people in the population that is always quoted for other cohorts, for other international groups in other countries, did that really apply to New Zealand? And I'll show in the next couple of slides some really great work that Maisie did. So Maisie's here in the audience, showing that our rates are higher than elsewhere. We also know that motor neuron disease is a disease of both genetics, because Some people with motor neuron disease have a family history, but for the majority, this disease strikes out of the blue, which is, really terrifying. You get no advanced warning. So we know that there's an interaction between environmental and genetic factors. And Kerry didn't present this, but this comes out of her study, that we think that in New Zealand our rate of familial cases is about 6%, and we're still at low numbers of people from whom we can gather that information. Those numbers might change, but we think we fall kind of in the same realm as international studies. And I'll just say here that, of course, amyotrophic lateral sclerosis is the major subtype of MND, and so on some of my slides I might use ALS. I won't go over this too much because it looks a bit complicated, but it's really not. And this is some nice work that Chris's colleague, Emma Al-Chalabi, who will come to New Zealand at the end of the year, has done. And he's done some really nice work showing that motor neuron disease, sorry, I'll just help part of you away there. Motor neuron disease is a disease of aging. We don't see people develop this disease in their youth, but the reason for that is that they need the accumulation of triggers, basically, over their lifetime, where some of those triggers will be genetic and some of those will be things they're exposed to in their environment. We don't really understand very well what those environmental triggers are, although there has been a big study done by our colleagues in Massey University. Some of you might have been involved in a big survey they did looking at environmental and workplace factors that were common to people who have MND. So we think these triggers might be cumulative, and that you will reach a point where you are triggered to develop MND and then manifest the disease. This looks a bit complicated too, but again, it's really not. Just to say that some of those triggers can be genetic, and our study is going to probe this a little bit more, because we're going to look in people who have a family history of disease, but also people who don't have a family history of disease, and whether there might be some genetic factors that have been part of their accumulation of risk that they have had over their lifetime before they develop the disease. And Chris knows this very well because he's discovered a number of these genes. But we know that for people who have a family history of disease, we might be able to find their gene in about 70% of those cases. And it might be true that for a few people who have sporadic forms of the disease, they might have a mutation too that we can identify. This is my last kind of intro slide before I talk about our own work on motor neuron disease that's really specific to New Zealand. And this is getting a little bit more into the nitty-gritty now. So if there's an interaction between genetic and environmental factors, what does that do? What are these triggering? And we know that the genetic and environmental factors can increase the tendency for neurons to build up and accumulate clumps of protein. I'll show that down here. You might not have seen an image like this before, but what it is some motor neurons. This is one motor neuron here and another here. This is from a person who had motor neuron disease and was a brain donor to our brain bank. And we stained their motor neurons for a protein called TDP43. And this neuron here, that's what we might expect to see in someone who doesn't have MND, where this brown stain here is the nucleus. That's kind of the powerhouse of the cell, where your DNA lives. And TDP is generally found in that part of the cell, and it can come in and out of that part of the cell quite happily. It's not impaired in any way in its movement. But in a neuron where, we're losing battery on that one. In a neuron where TDP is not looking normal, you can see this big clump here of the protein. It's no longer able to do its normal job in the nucleus, which is the powerhouse. It's trapped out here in the cytoplasm. And that's a really kind of characteristic feature of what we see in motor neuron disease, and we think that's what causes neurons to get sick and die. And what we're increasingly appreciating is that the motor neurons have this problem within themselves, but there are other cells in the brain called glia, or glue. We used to think they were pretty much useless and that they just glued the neurons together in the brain, but we now know that's not true. So these support cells, or glia, are actually extremely important, and they can also accumulate clumps of TDP and other proteins. So you can see up here an astrocyte and a microglia. Those are glial cells. And the final thing that we're also increasingly appreciating is that the blood vessels that kind of form a tree inside your brain and help deliver nutrients out to the brain tissue, help to take away the brain's waste, that tree of blood vessels should normally keep blood within the vessel and not let blood or any other factors inside the blood leak out into the meat of the brain. So there's a barrier there called the blood-brain barrier, but we know that that is also kind of broken in motor neuron disease. So all these features are starting to add together, where the neuron itself is accumulating clumps of rubbish, the glial cells which should be supporting and looking after the neurons are no longer doing so, and the blood vessels which should be kind of keeping their blood cell and other components internally are leaking them out into the area around the neurons. So, oh sorry, we're going a bit fast now. There we go. So now we know kind of the background of how my team thinks about motor neuron disease, and hopefully this is how the rest of the field thinks about the disease. Please meet my team. You can literally meet them because many of them are here. But we're very lucky to have great support from Professors Draganoff and Fall, and then my team really work right through from cells growing in a dish to brain tissue to brain cells, and now increasingly we're going to be looking at genetics and then into things like working with you, people affected by motor neuron disease, to learn more about your disease. And I've mentioned our stakeholders down here, where we've got Kerry from the Registry here. Obviously this is a Motor Neuron Disease New Zealand event. The Neurological Foundation Brain Bank, that's how we do our work, because a lot of the work on the brain tissue we get from the Brain Bank. And then we have great international collaborators. So these are our focuses. I'm going to go through just 4 focuses, and each of them has about a slide each, so it's not going to be too long. One of the key things is understanding motor neuron disease in New Zealand specifically. There's some great research being done internationally, but there's only one group that's focused on what motor neuron disease looks like here, and that's us. So we need to really understand our disease here before we can connect to that understanding internationally. Also, we look at how those toxic protein clumps accumulate, how we can clear them. We look at that blood leakage, both in tissues and in the cells that should be supporting that barrier. We're trying to identify new drugs, and the last thing obviously is studying genes, which some of you here might even be going to enroll in our study to look at the genetics of motor neuron disease. So first, I'll just tell you a little bit about a study that Kerry mentioned and I've already alluded to, and Maisie is here. We sought to understand whether New Zealand has the same rate of motor neuron disease as the rest of the world. And there have been some hints. A lot of you may have seen news articles over the last few years looking at clusters of motor neuron disease in different towns. Nelson, the ports of Nelson, there was a lot of talk about whether the fumigant methylbromide might have caused a cluster of, yeah, people are nodding. I don't think it's true that methylbromide increased people's risk of developing MND, but it got us thinking, are there hotspots of MND developing across the country? And even if we look nationally, what are our rates? So the best way to do this was not to look in little parts of New Zealand at small numbers of people, but to take the largest data set we could, which was the national statistics data on death certificates. So when people pass away, their cause of death is recorded. And when we look at the causes, the people who died from motor neuron disease, we can compare our rates to this exact same type of data from other countries. So by doing this, we actually looked at 2,264 people, which we'd never be able to do. We're doing a kind of regional study. And I won't kind of go over these graphs too much, but the point is that the graph is jumping around a bit, but overall you could draw a line that was horizontal. So over time, there's not an increase in our rate of motor neuron disease in New Zealand, which is good. We've accounted here for the aging of the population. We know that this is an age-linked disease. So as people get older, their risk of developing motor neuron disease increases. And so as the whole population ages, then we will expect more people to have MND. But when we take that out, there's no other increase in risk. So this is a bit different from what some other countries are showing, where they think that there is an increased risk of developing MND over time, independent of population aging. But what the take home from the study really was is that if we compared our rates to those of other countries using exactly the same kind of analysis, we actually have really high rates. We looked at 2 different time points. The first is here, and this is us here, where we had slightly different ways of collecting data about who died from motor neuron disease. And they give us slightly different answers, but either way, we've got much higher rates than other comparable countries, where all these other countries have similar health systems to ours. So we got a bit of press around this. You might have seen in the newspaper. Study reveals shocking death rates, and I did wear all my safety equipment for that photo, which is the most important thing according to the university. That is a plastinated brain, not any of the fresh brains that we treat very delicately. I was on, I think that was 7 Sharp, the project, and this was the one that, this really hurt me, to be honest. New Zealand is hard hit by cruel motor neuron disease, and scientists don't know why. So I felt seen by this. Another study actually came out quite soon after our study, which was a global motor neuron disease study, and it was not surprising that they came up with the same conclusion that we did, that New Zealand has really high rates of motor neuron disease. Unfortunately, there was some pretty bad press released around this that was, I think, quite confusing to people, where the headline was, "Shocking New Zealand death rate for motor neuron disease is 5 times the global average," because now the comparison is being done with third world countries, with countries with developing health systems where access to neurology is poor, we don't have good ascertainment or understanding of who actually has motor neuron disease. So it's comparing apples with oranges. I don't think it's fair to say that our death rate is 5 times the global average. I do think we're high, but that's not a take-home message that we would want to be the major one taken away by the community. So the next study I'll talk about, Kyra's here in the audience. She does this as her kind of daily experimental work to look at toxic protein clumps in human brain tissue. We're so privileged to be able to work with brain tissue from people who we interact with while they're alive, living with the disease, and after they pass on, we're able to explore what has happened in their brain. And we found that, not that surprisingly, people who die of motor neuron disease in New Zealand, their brains look pretty similar to the brains of people who die in other countries. But we have found some interesting things. I'm not going to get into that too much. But the major thing is that we see clumps where this brown guy here and these little stringies are that TDP43, the protein I talked about that should be in the nucleus, and now it's clumping in the cytoplasm. And we can even do this brain staining to give us kind of a genetic result, because there are some clumps that only come out of certain gene mutations. And there are some really cool, beautiful clumps. We have to take some pleasure in this really tragic and horrible manifestation of disease, but actually we see quite gorgeous things down the microscope, where here is the nucleus of a cell, that's that powerhouse I talked about, and we've got a clump here where it's a bit like a Jaffa. We've got this chocolate center and a crunchy coating, and we can identify the 2 different components of that. And that tells us a little bit about how disease developed, what happens sequentially. Even though it's at the end of life, we can kind of learn a little bit about what happened dynamically in that cell during its lifetime. Okay, we're getting there. This is referring back to that earlier slide I talked about, saying that it's not just about motor neurons, but it's about, we haven't developed effective treatments when we're just thinking about this disease as a disease of motor neurons alone. We have to be thinking about the entire picture. So what we do, and this is kind of one of the most unique and perhaps shocking aspects of our work, Charlotte in the audience and Saray, and actually Maisie as well, are growing cells from the brain tissue that we receive from people who've passed away from motor neuron disease. If a brain comes to us reasonably fresh after death, we can actually take a little piece of that brain, break it into pieces in a dish, and the cells will continue to grow. Just a few of the cells, a certain type of cell, Those are the cells of the blood vessels, and actually there's a list of the cells that we can grow down here, but the ones that we mainly work with are these pericyte cells that are blood-brain barrier cells and that are involved in brain inflammation. So we can literally have disease in a dish, a little kind of microcosm, a picture of someone's disease growing in a dish. We can pop drugs on that and see how they respond. We can look at how the proteins are clumping or not. It's an amazing and quite stunning model to work on. And we can use these cells to screen for large numbers of molecules that might affect the way these cells are behaving in an inflammatory way or in terms of the protein clumping. This is some work that our postdoc Deidre McVeigh did, and basically what she's looking at, I won't go over this too much, but she stimulates these pericyte cells in a dish and she makes them inflamed, they're angry, they're releasing these molecules that could damage neurons, and then she's popping drugs on them that will decrease, kind of dampen that inflammatory response. The idea is that we might be able to take these forward to quieten down that inflammatory phenotype we see in motor neuron disease. And the last slide I'm going to talk about, which some of you may know a little about, referring back to that genetic slide I presented at the beginning. Could genetics underpin our high rates of motor neuron disease? We really want to understand if our genetics here in New Zealand is different from what we see in other countries. And we also want to recognise that the people involved in this study want to find out for themselves what is happening and what does it mean for their family. There's actually at the moment no way for a person with sporadic disease to find out about their genes. You can't go through the public health system and have a funded and supported answer about your genetics. So through our study you can do that if you want to. The great thing is we also don't insist that anyone learn their genetic result at all. You can be part of our study and not learn anything about your genes if you don't want to. So we are going to work with 300 people with motor neuron disease and 30 controls, collect blood, and we're working with colleagues in Australia and with Chris's group to find those known genes that we already know cause disease and to find novel new genes, get new chances of finding therapies. So the summary of my talk is that the time is right for more research that's really specific to New Zealand. We need to know what differs, what's the same, how we can benefit from those international advances that Chris will tell us more about. And I think Claire's given just a beautiful picture of how New Zealand research is going to advance with MND at the helm. We know that the way that the brain tissue, that the genetics, and the way that people present with disease are all linked together. And we also know that it's not just about neurons. They're the most important part of the disease, but we will get to a point where we've solved the problem that neurons have, and we need to think about those glial cells. We need to think about what the blood vessels are doing. And it may be that therapies that are not extremely effective for neurons might work well with other therapies that target glial cells and blood vessels. They might augment and work together with one another to give us a therapy that really cures disease. And the nationwide genetic study, I think, will really inform our future work. So I think we all know what these building blocks are that are going to get us to the point where we can cure motor neuron disease in New Zealand, but I want to just acknowledge all the people that are involved in these different parts. We've got Lydia here in the audience, who's our new manager of the research network, and Chitra is the person who you interact with if you're part of our genetic study. So thank you to everyone who is part of this, and it's just such a pleasure to work in such a motivated team of people and to work with a community who come along to events like this. You know, you give your time freely to come and listen to what we're doing, and it's a pleasure and a privilege. This is all the team, and I have to also acknowledge our very generous funders. The New Zealand government is great, but it's families and organisations who have a special interest and who are affected by motor neuron disease who really give us the transformational dream funding that's not kind of locked into different ways of spending. We can really use that in a way that pushes the research forward quickly. Thank you very much.
Thank you, Emma. We all agree that was fascinating for us non-scientists in the building, and thank you for making it so we could all understand. It's just, it's just so, it makes me feel quite intelligent when I listen to you presenting. I thought, oh, I understood that. But it's, as you said, it's such an exciting time for motor neuron disease research, and I think those building blocks that you talked about are really important, about everyone working together and everyone playing their part. So thank you. And then we'll be on the panel later, obviously. So now it's afternoon tea time. Okay, today is obviously advertised as an update on MND Research, Homegrown and Abroad. So it gives me great pleasure to introduce our next speaker, Professor Chris Shaw. Chris is Professor of Neurology and Neurogenetics at the Institute of Psychiatry, Psychology and Neuroscience at King's College London. He's also head of the Department of Basic and Clinical Neuroscience, Director of the Maurice Wall Clinical Neuroscience Institute at King's College London, and an honorary consultant neurologist and neurogeneticist at King's College Hospital. His major research interest is in the genetic, molecular and cellular basis of motor neuron disease. Research led by Professor Shaw has identified mutations in many genes causing ALS and MND, including two of the most common, TDP and FUS. And Professor Shaw conducted his clinical training in general medicine and neurology in New Zealand. So, welcome home. Thanks, Chris.
Thanks, Carl. I tried to put in as many tricky words as I could. Indeed, this is Homegrown and Abroad. I am homegrown and living abroad for the moment. But I'd like to come back here on a regular basis, and I was absolutely delighted that Emma came to work with me 10 years ago. Sometime. We were just talking about aging before, but we won't age ourselves too much. And then, against my good advice, she came back and said, oh, it's really hard to set up research in New Zealand. And I see an extraordinary team of young people, smart young people here, that she's supporting the research of. And to hear the extraordinary work of Claire and other colleagues in building the momentum to build research that's actually homegrown in New Zealand is really exciting, and it makes me very proud to be a New Zealander and to be part of this community, so I'm honoured to be here. And thank you very much for coming to hear me speak. So, obviously, are we there yet? That's a very annoying phrase that children in the back seat might say as we're driving somewhere. And I'm going to talk about gene therapy. Just my own little story about becoming involved in MND research was that I actually started off as a multiple sclerosis researcher in Wellington, and I wrote some papers and managed to get a fellowship to go to Cambridge. And while doing clinics in Cambridge, I came across a woman who had motor neuron disease. She diagnosed it herself because she recognized the symptoms that her mother had, her aunt and her uncle had. And up until the time that I met her, I didn't know that it could actually run in families. So that was pretty shocking. And she told me how she'd spent so much of her life waiting for this to happen. And I thought, well, this is a really challenging disease, but the fact that she knew what was coming made it even harder. And then, of course, she had the realization that she may have passed this on to her children, which is like extra difficult. And then a month later, I met another woman with exactly the same set of problems, and she'd had a family history as well. So I searched around, and there was a lovely chap in King's called Nigel Lee, the Emma as well, and they just discovered the SOD1 gene, and they both happened to have SOD1 mutations. And I thought, wow, this is amazing. And I, oops. Has it died? Okay. And he started working with me, and in the end he offered me a job to go down to King's, and I've been there ever since, 25 years, working on exactly the same problem. And I was under the illusion that, having discovered SOD1, we'd have it all sorted very quickly, and there'd be some fantastic therapies. Well, I think we're just about there, and that's what I've come to talk to you about today, because we are at the cusp of something really extraordinary. Okay, where are we? So I'm going to talk about motor neuron disease. This is Patrick the Optimist. Those of you who don't know his contribution, there is a connection with frontotemporal dementia. It doesn't always accompany the disease, but I'm going to show how the connection between these two conditions is linked both in terms of pathology, the proteins that accumulate in the brain, but also genetics. I'm going to talk about the genetics of familial motor neuron disease a bit, and then I'm going to talk about a different motor neuron disorder called spinal muscular atrophy, and it's genetic therapy in the context of spinal muscular atrophy that basically is giving us a big kick in the bottom to get stuck into doing gene therapy for motor neuron disease. So we're learning from those colleagues. So this is the chap who was the first person to point the finger at the motor neuron. This is a muscle wasting condition, and for many years it was thought to be just a muscle problem, but actually what he saw was that the motor neurons didn't look normal. And he actually spotted the accumulation of protein inside the motor neurons, and he realized that this was probably significant. And you can see that little aggregate there. It's very similar to the ones that Emma was pointing out before, and this one's really stuffed with protein. And he knew that that was a sign of a sick cell. It took us 160 years to actually figure out what the protein was, and it turned out to be TDP-43, but that was a really important start. So he named it this terrible name, amyotrophic lateral sclerosis. So for those of you who'd like to hear and understand words, I'll go through it a little bit. So if you look at the spinal cord, you can see these dark grey areas that he's drawn. Those are scars, and in French it's called sclerosis. And it's on the side of the spinal cord, which is the lateral part of the spinal cord. And the amyotrophy is basically muscle wasting. So you put this all together into a terribly difficult and garbled phrase, which is amyotrophic lateral sclerosis. So ALS is so much easier. But like Emma, I slip between ALS and MND. In fact, they are pretty much the same condition. So I apologize if that's confusing. There was a chap called Lord Brain, that's his real name, who was a neurologist, who knew he was going to become a neurologist, extraordinary. And he wanted to call it motor neuron disease, which seems like a very sensible thing to do, but it hasn't really caught on in the rest of the world. He was actually the editor of a journal called Brain. And the person before him, who was the editor, was called Henry Head. So anyway, that's a lot to follow on from. But in America, you hear it called Lou Gehrig's disease, after a very famous baseball player who had this condition. But the description that I think is most telling was one that I came across in the death certificate from the late 1800s, which called it the creeping paralysis. Because this is very much the way disease starts, which you all know. It starts in one part of the body, creeps to other parts of the body, and causes paralysis. And I thought that's a very evocative name. So we've had this fantastic discussion, and Emma's actually shown that New Zealand, for reasons we don't fully understand, but are about to understand because of the important research that's going on here, New Zealand's got a really high incidence of this, and that's a concern. If you're a man, your lifetime risk is about 1 in 300. This keeps fading out, I don't know what I'm doing. And a woman, it's about 1 in 400. So it's not that rare. It's only rare when I'm writing to a rare diseases charity, in which case it's very rare. But otherwise, I'm saying actually this is a really significant and important disease. And most of us know 300 men and 400 women. And we're going to know somebody, people off the street. So, and because motor neuron disease is such a striking condition and challenging condition, I think it really does touch people. And I think I really admire the way you guys have harnessed that pretty much neglected pool of response, emotional response to this condition that people have experienced over the years, to say we need to do better than we are currently doing it. And I think it's fantastic. You're going to get all the clinicians in a room with a big stick, and you're going to get them to behave and start to standardize their care and really think about what people with motor neuron disease need. So hats off to you. Unfortunately, I'm out of the country, but anyway, so just talking, not acceptable. I'm coming back in December to your big conference. I'm glad you put me up there. I was feeling left out. So there are connections between motor neuron disease. I'm not going to go through it in great detail, but there's sometimes people have some subtle cognitive change. It's not dementia, but it's a really subtle change in terms of word finding and some planning. And we know that even we think about the spinal cord as being the source of the disease, but we do see pathology, the TDP-43 that Emma talked about in other parts. And interestingly, all the people that were working on frontal dementia, which is a condition that presents with some behavioral change and some personality change, those people also have problems in the spinal motor neurons as well. But because the doctors who look after dementia are different from the doctors who look after neuromuscular diseases, they don't really talk for a long time. And it took us a long time to really start to put these things together. And why is that important? It's because it really helps us understand the disease. It also means that treatment that you might find for one condition may work for the other, and that is a really important and powerful advance. Okay, so these are some of the genes. SOD1 we don't really think is associated with dementia particularly. TDP43 can be, and these genes in the middle, ubiquitin 2 and C9orf that Emma talked about, they can definitely cause one or the other. And then some of these others are much more likely to be frontotemporal dementia. But we do now consider this to be a spectrum, and some people are unfortunate enough to get a bit of both. So that's where we are. And if we were to then look at all the brains and see how many people had different sorts of pathology, you could see that in motor neuron disease, most people have this TDP43 protein accumulating, and a very small number have the FUS protein, and a very small number have the SOD1 protein. But we know that those people will have gene defects. So if you do a gene test, like Emma's doing, and you say, okay, this person doesn't have a FUS mutation, doesn't have a SOD1 mutation, then we can be almost 100% certain they've got TDP43 accumulating. It could be from lots of other genes, but that is the common pathology. And interestingly, in frontotemporal dementia, about 60% of people also have TDP43. So that's, it's a powerful step forward. And then this is some of the pictures. We can see TDP43 accumulating in the motor neuron, but also in the frontal and temporal lobes, we're seeing TDP43 accumulating. And we know that the signature of pathologically misbehaving TDP43, it moves from the nucleus into the cytoplasm, it's cut up into smaller fragments, it becomes sticky and insoluble. That's part of its problem. And there are molecular tags that Emma's and others have helped characterize to say that this is in the wrong place and doing the wrong thing. The cell knows that, but it can't get rid of it. So if you like, it's the rubbish in the cell is accumulating and it's unable to clear it. So that protein recycling is failing. Okay, I apologize. Emma has seen the slide 100 times because I put it into every single talk I have ever given for the last 20 years. And it's a really old slide. And this is a kind of old picture of DNA, and you can tell this is a real slide because I used a font which was shadow, which was really cool. I kept shadow. But the idea was that we could understand the genetics. We could then model the disease in cells and animals, and we'd get a better understanding about what the process was. And we'd also have a tool to be able to test drugs to see whether we can slow down, stop, or reverse that process. So that was the strategy. I've been gene hunting for 25 years. We've discovered one or two. And we have made enormous progress in understanding disease mechanisms. That is a really exciting advantage. I'm going to talk about all the different genes. So the balls in green are those that are associated with frontotemporal dementia, FTD. And the blue and red balls are associated with motor neuron disease. And the size of the balls reflects the size of the population affected with either particular gene defect. So you can see the larger balls, SOD1 and C9orf72, and most of the others are pretty rare genes. Not many people have those conditions, but they have given us extra pieces of a jigsaw puzzle to figure out what pathways are going wrong. Right? When you're doing a jigsaw puzzle, you get some big pieces, you can start to figure out what's going on. These are all the little pieces that we can start to see and really see what pathways. This is really weird. Pathways are affected. So we've got 4 new genes just this year. We're trying to get a couple published at the moment. And these are genes that we've either been part of the first group to discover or part of a consortium, international consortium that others have contributed. We've contributed samples to that have led to these discoveries. And it just reflects how important it is to collaborate, international collaborations, to make this thing happen. And it's great that New Zealand is starting to collect a proper DNA sample to be able to be a part of this incredible activity, international activity. So how does this help? We've been gene hunting all this time. Well, firstly, something a lot of people want to know is, why me? Why has this happened to me? And if we can find a gene defect, I can say, I'm afraid this is the spelling mistake in your DNA, which you inherited, and that has caused this protein to misbehave, and that is why you've got the disease. So, that is actually an answer. It's a real answer. And that's been where we've had to leave it, because there was no actually follow-on from that. Say you've got this particular gene defect, then we can give you this particular treatment. But that's about to change. So the other group of people are people who've got no family history at all. They're not really worried about genetics. And if we know all the really strong genes coming through, right, we could test them and say, well, you don't have one of the strong genes, so your kids probably are not really at risk, and that's a great relief. So we can give people some positive news and say, we're not too worried about the next generation. And then occasionally we can do something a little bit more unusual, which is called pre-implantation genetic diagnosis, which is basically in vitro fertilization, IVF. So the sperm and the egg are fertilized in the laboratory, and the embryo grows to about a 40-cell stage, and then they take a cell off, and they do a gene study on it, and they figure out whether it's got the gene defect that we know one of the parents has, and the embryos that don't have it, we can put those back into the womb, and hopefully a baby will result. And indeed, this little chap did in 2013, and his name was Thomas, and I looked after his grandmother, who died of this disease, and sadly his uncle died 2 weeks after he was born, but he did see him. So he will not get this disease, right? Because we tested him before he was put back into the womb, which sounds like a strange thing to do, and it is a small thing, but it is something. It's something this family are fighting back against the disease, and this is what we're able to do. Anyway, just about 6 months ago, we got the license to do this for every single gene for motor neuron disease, which was a great step forward, because every gene we discovered before that, we had to apply a license, which would take 6 months to get a license for every single gene. It was a real nightmare, administrative nightmare. Anyway, that's on his 6th birthday, so they sent me that recently, which is really nice. Yeah. So I feel very cool about this, but it's a very small thing we've done, and we need to do a lot better. Bit of a biology lesson. I apologize for this. Actually, I don't apologize for this. We should all be thinking about biology. And this is a cell which is sort of broken down. This is the nucleus, that's the cell, and this is the DNA which is wrapped up into something like a woolen ball, right? But it's a single strand of DNA. And these are the bases, the letters in the genetic code, GATC. So you can't really use that because it's sitting in the nucleus, and the protein-making machinery is out in the cytoplasm. So the cell has to make a little photocopy of each gene, which then gets pumped out into the cytoplasm, into the protein-making machinery, and all the amino acids are stuck together like beads on a necklace to make the protein. So that needs to happen. So these are the amino acids in yellow that are coming out. This is the photocopy, which we call messenger RNA. It's a messenger because it's taking the message from the DNA out to the protein-making machinery, and that's how we make every single protein in your body, and you are entirely made up pretty much of protein and salty water. A bit of lipid, fat. Some of us a little more than others. So if there's a mutation in the DNA here, that translates to a mutation in your RNA, and that can cause a mutation, a change in the amino acid. Just a single amino acid changing can change the shape and behavior of that protein, so it starts to accumulate like TDP43 or like FUS. And it's tragic that such a tiny little mistake has such a big impact, but that's the truth. So mutant or damaged proteins accumulate inside motor neurons, and motor neurons seem to be quite poor at clearing them. We don't fully understand it, but we know that the neurons you die with are the ones you're born with, and they are enormous in their size. They're bigger cells in the body by a very long way. We look at them down the microscope and say, well, that's a really big cell. But that volume that we see there is 1/1,000,000th of the volume of that cell. And when I'm lecturing to science and medical students, I say, if I'm a 6-foot tall motor neuron, right, and I'm sitting in the spinal cord, and I want to activate a muscle in my foot, my axon has to reach out to Paris from London. It's a huge distance, right? So you've got to keep that going all your life. So that cell has to be fantastically tidy. My mother's in the audience, she'll know that. I've learned to be tidier. I'm pretty tidy now, except my desk at work. So it has to have fantastic housekeeping skills to keep that cell functioning. And a lot of the functions that we imagined all happen in the nucleus are actually happening way out in the periphery, sort of slightly out of sight. So this is just a badly drawn neuron. Somebody described it as a squash frog. So that's the cell. There's the nucleus, and that's where TDP-43 mostly sits. It's in the highest concentration there. But actually, it starts to cause a problem when it accumulates in the cell body. And what happens is it starts to stick together. This is the black thread, and it gets labeled by ubiquitin and P62. Those are basically tags that the cell says this protein is damaged and needs to be destroyed. And the cell tries to truck it off to this structure here called the proteasome, which is like the incinerator in your sink where you're stuffing vegetables but no hard objects down your incinerator to break them up and to recycle the protein back into amino acids that you can use again. Or if you really want to destroy them properly, you send them off to this green blob here, which is the autophagosome, and that's able to degrade not only big aggregates of proteins but also parts of the cell that are damaged, whole organelles within the cell that are damaged. So those are really, really important pathways. And if you look at all the genes we've discovered, I can start to map them out on the cell as to where they're getting things wrong. So the first thing is if your shuttling in and out of the nucleus is damaged in some way, these genes might lead to the accumulation of TDP-43 out in the cell. And then of course if your shuttling down this really long axon is failing for some reason, these genes relate to axonal transport, then TDP-43 is a really sticky protein. It's like chewing gum, right? And starts to cause problems in the axons and starts to accumulate. And then the biggest hit from all the different genes, and these are all different gene names, you've heard about ubiquitin too, they all relate to the protein recycling function of the cell. So you can hit that in lots of different ways, and guess what? Chewing gum, TDP-43 starts to accumulate and starts to damage the motor neurons. So it's a really, really sticky and difficult protein, and from all these different problems in the cell, it's TDP-43 that seems to accumulate. There's also a story around mitochondria, which is the energy source of the cell, the battery of the cell, and when you haven't got enough energy, it's harder to clear these things as well, so we think that's how it's working. But these are all the different aspects. So, this is like 25 years worth of work to put all these little pieces of the jigsaw puzzle to say, okay, we've got some understanding. And Emma and lots of colleagues and I have been working on this for a very long time to try and understand disease mechanisms. We then work up ways of reading these out, these things that are happening in cells, to say, okay, this is where it's going wrong, how can we improve that particular function? And we can do some in-house testing in some of our fancy machines. We can work with industry, we've got big robots and lots of money. And the idea is we get some compounds, these are chemicals that are drugs that we might be able to use. We take them back to our animals. This is a fruit fly, a zebrafish, and a mouse. And that in the virtuous circle, in the perfect circumstance, the ones that really work in the animals would be brought to man to be tested. Now, I've been doing this a very long time, and I've been trying to work with industry for a very long time, and this circle doesn't work very well, right? So we've got right around to this stage where we're doing, compound discovery, and then somebody comes in and cancels the whole program. We spent years doing it. Very, very depressing. So we're starting, so where have we got to in all these years? We've got Registry, and curiously, New Zealand was one of the last developed countries in the world to make this available to patients on the health service, which is criminal. And I think it wouldn't happen again because of people like you, Claire, who would be down to the health minister, right, and threatening him with some dire consequence. And I think it was really because in New Zealand we weren't organised enough to really put pressure on the government to make this a funded drug. It does work, but the effect is modest. It's not enough. So the new drug, which has been licensed in the United States and Japan, called Dementia, it's quite a challenge because you have to have it intravenously every day for 14 days, and then you have 14 days off, and then you have 14 days on, and that is a really big commitment. So there are companies that are now trying to make an oral tablet, which hopefully will be effective, but it's still a pretty modest effect. We've not made the dramatic change that we really would like to see. So this is my new slide with a new funky font. And that's to say, okay, can we take these gene discoveries and actually be much more direct and turn them into therapies themselves? And when you make a gene discovery, what you do is you knock it down, see what happens when that's missing, or you overexpress it and you try and say, well, what's happening with the healthy version of the protein versus the mutant version of the protein, what's going wrong? And gene therapy is actually knocking genes down that are toxic or supplementing them when they're deficient. And I started a collaboration with the guru of gene therapy about 3 or 4 years ago. And we had all these plans, and he liked all my ideas. He then ran off with all my ideas and has raised his own company and patented all sorts of things, which is pretty unfortunate. And when we discovered this, one of my senior colleagues and I came back and he said, we should do this ourselves, which we're going to do. And I'm going to talk a little bit about that. So we are going to try and generate gene therapies that really hit hard at the disease. My problem with small molecules, a lot of work's gone into small molecules, but I think they're going to have small effects. Gene therapies is like taking a shotgun to the process. Okay, so how the gene therapies might work, this is the whole sad story about mutations from DNA going to RNA, making abnormal protein. Can we knock out the middleman? So the idea is people are generating these tiny little fragments of DNA which are able to bind to the RNA because it's a perfect match. And when the cell sees that, it sends an enzyme out, Pac-Man, and is able to eat up the RNA, but it leaves the DNA there, which can then go and bind to another RNA. And these are antisense therapy trials. Okay, is this science fiction? No, it's not. In fact, there are members of this audience who have a sister who's a patient of mine, a New Zealander, who's living in London, or living in England, and she's been in a trial for the SOD1 gene mutation. And she has not changed in terms of her disease progression over the last 3 to 4 years, which is absolutely remarkable. And I'm a consultant to the company that's been working on this drug, and I went to a presentation in December in which they told the story of people who have a very super aggressive form of this disease. They have a very specific mutation. And this mutation usually causes death within 12 to 18 months of symptom onset. And indeed, they had 4 people who were given the placebo, and they had that disease course. The 4 people who were on the active drug did not progress, and some of them got better. Astonishing. So this is for one gene, for SOD1. But it was incredibly moving experience to be in the room to hear that data for the first time. I'm a lovely patient who I've known all these years. She's got pretty slowly progressive disease, so it's hard to see a change, and maybe she just had plateaued and I've been seeing it all this time, but actually hearing that news gave me incredible hope. So this little Pac-Man and this little combination with the antisense DNA is really making a difference. If we can do it for one gene, we can probably do it for others. So Ionis are the brains behind all the science, and Biogen is the big pharma which has come in to invest to make these trials happen. They did a trial and therapy for spinal muscular atrophy. I'm going to talk about that. That's an infantile and childhood onset disease. I'll talk a bit about that later. But they've got the SOD1 trial, they've got Huntington's trial, Tau trial for Alzheimer's disease. And there's a trial we're starting very shortly in the UK on a gene which is present in New Zealand quite frequently, C9orf72. If it works for SOD1, it'll probably work for some of these other genes. I already know colleagues working on Huntington's showing it working there. I think there's a trial for patients here in New Zealand on that Huntington's trial. So yeah, so it's incredibly exciting that these therapies are not just going on in America, right, but they're actually going on in New Zealand. So yeah, I'm very proud. Okay, back to this picture. Oh, what are we doing? Yes, so this is our strategy. So I don't make antisense oligonucleotides. We're going to try and use a different technique. One of the problems with antisense nucleotides is you have to keep giving them all the time. Now the drug companies like that because they make them and they can sell them, and it goes on and on and on. But we've got a different strategy, which the Americans have ably called one and done, which is to use a cold virus to deliver the gene that's missing, or to deliver a gene that will lock down a toxic gene. Okay, I'll explain how this works in a minute. But these are the targets. We're going to try and supplement a gene that's missing in frontal dementia called progranulin. We're going to try and knock down the big nasty C9orf72. We're also going to try and alter a pathway, which is a gene called ataxin-2. And then we've got a wildcard experiment where we're trying to turn on the recycling and protective machinery of the cell to try and help clear TDP43. So that's our strategy. I can't talk too much about it because it's all very secret. Well, not so secret now. But this is how it works. So that cartoon is a virus. It's a tiny, tiny virus. And people get exposed to all these viruses all the time. And obviously they're there to infect you. But what we do is we take out the viral DNA so it can't replicate and it doesn't do you any harm. And we put back our own little secret package, which carries either the gene that's missing or a little sort of antisense microRNA, which is able to knock genes down. We're not giving a drug after that, we're just giving one injection. It's very cool technology. At the moment, we're doing this in mice, but we plan to do this in sheep fairly soon, and we hope to do this in man in the near future. And these therapies are going on, and they are very likely to work. So we're very excited because the results we're seeing in mice look very promising. Okay, so this is what we've done over time. We've tried to figure out exactly how we can make the very best sort of virus to take this into cells. It is able to deliver the gene to the most number of nerve cells, and that has the most powerful effect of either supplementing a gene that's missing or knocking it down. It takes quite a lot of science and investment and time to do this, but we are in the process of doing that. And what's amazing is that some of these viruses really love motor neurons. You know, you can inject them into the blood and they get into your motor neurons. So we are very optimistic this is a good place to start. Okay, so we can also get neurons, genes delivered to neurons throughout the brain as well. I won't go into that too much detail. So now I'm going to talk about spinal muscular atrophy. So this is an inherited disorder that babies get, right? Infants. They're often born floppy, and these children, sadly, they never get strong. They never sit, they never stand, they never crawl, and they usually never talk because by the time they reach the age to speak, they've got a tube in their throat and they're ventilated. Really, really harsh disease, but it's a motor neuron disease. And they're missing a gene called the survival motor neuron gene. And you can see I've done that very crudely here. That whole gene is missing, it's deleted. And that means the RNA is deleted, and that means instead of having normal proteins, you can see the red stain here in the motor neurons. That's the normal protein, and it's missing in somebody with spinal muscular atrophy. So they're missing a gene that makes them miss a protein, and that's why they have this devastating, fatal motor neuron disease. Okay, so life without survival motor neuron disease, so survival motor neuron protein, means you end up in a wheelchair, you're on a ventilator the whole time, and you might have a little bit of muscle movement. Well, Brian Casper has been working on this for quite a long time. And he was working with the same viruses that we're working with, and he managed to put in the survival motor neuron gene into that little virus, and he gave an injection to these kids between the ages of 1 and 7 months. He gave it to 15 children, and none of them ended up on ventilators, and some of them are running around, right? One of them is speaking 3 languages. It's almost a normal life from a single injection, given once, at the right time, to the right people. So we think this is time for us to really do something for motor neuron disease. This is another motor neuron disease. Why can't we do something similar here? And if you don't believe me, hopefully this will work. I'll try and get that going. So this is a video of a child who had this injection at 2 months of age. And it's actually taken from YouTube. because somebody videotaped. Now the company, I actually work for this company, Abexis, as a consultant, but they don't share out their videos. The videos they've got are even more impressive. But remember, these children never sit, they never stand, they never walk, and they never talk. Now, this isn't a completely healthy 19-month-old child. You know, you can see that they're a little bit unsteady. It's pretty amazing. And this is a month later, and these children are continuing to get stronger. So we're rescuing their motor neurons, and their motor neurons are becoming more and more effective. So I think that's actually remarkable, and that we should be trying to do something similar for people with motor neuron disease. Anyway, lots and lots of people have contributed to this work over a very, very long period of time, and this person may be familiar to you. She's down under. And this is the current team. And so I've gone from gene hunting, and you know, we can discover lots more genes, but are we really making a difference? Well, yes, we have in terms of our understanding, but am I helping people? Well, not yet. And now I'm not going to sit around waiting for small molecules to do wondrous things and for other people to discover them. We're actually going to go out and make therapies ourselves and get this to the clinic. That's the plan. So thank you very much for listening. These are all the people that I were working. I didn't have a gene therapy training 2 years ago, and now I've got all these people working on this program. So thank you very much.
Thank you very much, Professor Shaw. Travelling all this way just to talk to little old us.
With my mum, exactly.
And thank you to your mum for bringing him here. And make sure he gets to bed early tonight and tidies his room. Yeah, I feel my IQ has just risen hugely after that. That was just amazing, and it's, you know, it's such promising, you know, work. I mean, I'm talking to the researchers, how much progress has been made in the last sort of 6-9 years or so. It's just phenomenal. It's exponential. So we're just going to finish the afternoon with a small Q&A session. So we'll probably bring some chairs up to the front for you guys. Any questions from the floor?
Hold on.
Yep, just hold on there.
I'm sorry I forgot your name.
Me, Chris.
You, Chris the man.
You are.
I'm Sally James.
Hi.
Now, when these injections are available for people for once, so we go, Jerry, would that stop them just like this, or will it go backwards and
Yeah, so
it'd just be like this?
we don't have absolute proof of this, but the mice have been made in which you can switch on TDP-43 and the mice become almost completely paralyzed. And when we look in the mouse brain and spinal cord, we see TDP-43 accumulated. Now, if you then switch it off by a genetic switch, right, the mice return to normal function if you get them early enough. It's a really remarkable result. Now, half the motor neurons are gone, right, and you don't grow new motor neurons, but the motor neurons that were sick and not working are able to clear the TDP-43 and start functioning again. So they start activating the muscles they weren't able to activate before, and then they branch out to activate muscles that have lost their neurons altogether. So although they only have got half of the motor neurons, they're able to almost completely recover function. It's a really remarkable story. So I think if we were able to switch off the disease, there might be a population of neurons that would then come back to functioning. And so that's why I think for some of the patients in those trials, we're actually seeing some of them get better. And they have videos of people, you know, really having struggled to walk, but actually then subsequently walking up steps. You know, I wouldn't believe it if I hadn't seen it with my own eyes. And interestingly, the drug companies normally do very small trials of like 20, 30, 40 people, just to show that it's safe and that it might be a hint of an effect, right? And then they do a much bigger trial with hundreds of people with motor neuron disease. And then they can get a license to launch the drug and commercialize it. They're not doing that for the SOD1 antisense. They're going straight to launch it and make it available for people because the effect was so strong.
So when will it be available?
So that's a big question, but certainly in America, the FDA are looking at this. Now, I'm wanting to give you unrealistic hope. These drugs are being licensed and are very, very expensive. Right? In America, it's all about insurance. You've got insurance. You know, it's bad luck for the insurance company, but good luck for the person.
But you only need it once.
No. So those antisense therapies, they're giving by 3 monthly lumbar punctures, which is not a trivial thing, I understand. But, you know, that's a pretty extraordinary result. So, you know, my patient is really happy to go up and have her lumbar puncture because she has noticed incredible stabilization of her condition. The viral vector, the little virus, that is just given once. And that's a different process. Rather than just giving a small DNA chemical, we're actually giving a virus which is delivering the whole gene, and that gene will stay in those cells for as long as they're there. That's why it's a one and done therapy, if you like that phrase.
Can they get better too?
So that's not been done in man yet for this particular condition, except for the babies with spinal muscular atrophy. So that was a virus. And those kids are doing incredibly well.
You get yourself back to England, back to work.
I'm still working here, I can tell you that.
Thank you very much. Any more questions?
Hi, my name's Amita, and my brother's a bit shy, but he's I think I'm sure. Yeah. Okay, so my brother's been diagnosed within the last couple of months with motor neuron, but he had just been deteriorating with symptoms for over a year. But we missed the first couple of speakers, so our family's just kind of come to terms with it in the last month, and our situation's kind of tricky, which the MND organisation is aware of because I've touched base with them, but with the research and things like that, he's interested.
Great.
But because there is no cure, so we are looking at any alternative to see what will work, what won't work. You know, anything's possible. We're also people of faith, so prayers are all around. But like with the research, if we were to get involved with it, does that mean that we shouldn't try the alternatives because it might sway the research a bit? Like if we were to get involved with that, even with your research, you've got a Kiwi girl over there.
Yeah, great
With
question.
money figures.
Great
How
question.
how much would it cost to say get my brother if we were to raise funds to get him to be in something that you're involved with over there? So
I'm going to hand over to Kerry in just a second, but we want to connect people to research. The research studies that are being done in New Zealand are mostly genetics, which is the study I talked about. So it doesn't matter what anybody takes, what they're doing with themselves, your genes are going to stay the same. So anyone doing the gene study, nothing that you take or do is going to affect our result from that. That it might not necessarily be true for other studies, but at the moment we have no trials being conducted in New Zealand. But I'll pass over to Kerry now because she can tell you about signing up with the registry and the potential that we're going to have clinical trials here in New Zealand, or that people can be connected to clinical trials that are in Australia, for example.
So yeah, as Emma said, in New Zealand we are yet to have a clinical trial for any new drug for motor neuron disease. And although these clinical trials are happening, they're happening in places like London and they're happening in America. And so that is the purpose, largely, of the registry, to try and attract these things. And in the last few months, we have been approached by some pharmaceutical companies who are looking at the possibility of bringing them here. And so, thank goodness, we're really excited. We don't know if they will come, we don't know when they will come, but we're doing everything we can to try and make that happen. But yeah, please join the registry. If you'd like some more information about it, it's on the MND New Zealand website, and please do email me. In terms of trying alternative treatments, I think I'm not a clinician, but I would say that you need to talk to your doctor. In terms of treatments and research, that will be, if you do join any research study, it will be spelled out to you very clearly, exactly what things are allowed and what aren't.
So I'd say about 60 or 70% of the patients that I see are also taking alternative therapies. And in the early days, people were very nervous about saying it, and now we ask to say, yeah, what else are you taking? And often there's a huge list of vitamins, minerals, herbal treatments, and spiritual treatments. So we think that that's great, and it's a really positive thing that people are doing and engaging. There are occasionally, if you're on a drug trial, they want to know what other things you're taking, and some of them might interfere with that, so that would be a concern. But I think that's a very unusual circumstance. I hope that I haven't given you false hope to say I've got some secret, therapies that are working in London, and we're way ahead of the game. The standard of care in New Zealand is almost exactly the same as the standard of care in the UK, and almost exactly the same as the standard of care in the United States and every other part of the world, right? So it's not that you're really disadvantaged. Some of the more radical and extreme therapies that are being trialed with stem cell transplants are happening in the US and in Israel. I personally don't believe that they're likely to be successful, and I've been working in this field for 25 years and know almost all the people that are doing that work. I think it's good that they're doing the trials because we'll get an answer, but I don't have enormous hope, and they are telling me in my ear that it's really working. But we'll wait and see, all right? So those are the only therapies, the radical therapies that I think are out there that are being trialed at the moment, that people are certainly recruiting to. And I'm a bit worried that the trial, at least in Israel, they've stopped running it as a normal trial, and they're now just charging people to come in on compassionate grounds, compassionately giving them $50,000 to have a pretty unproven therapy. And so people are selling their houses, borrowing lots of money to go and get this one-off shot for a therapy, which I don't think there's much evidence to say it works. So I'm pretty deeply skeptical about their motivation, that they need $50,000 to do that. I would say that the gene therapy trials are coming, they're usually for people who've got a specific gene defect. And one of the great things about what Emma and Kerry are trying to do is to be able to offer gene testing to people and say, okay, you have got that gene defect, and we can put you into touch with the people that are doing those gene therapy trials. We've got some ideas, and I've been trying to tell the gene therapy companies to say New Zealand is a really good place to do this. Right? There's a sufficient number of people. There's a fantastic network of really clever, smart, active people who are able to mobilize patients to become part of those trials. So. New Zealand is definitely getting plugged in to the international scene. It always takes a bit of time.
Can I just add very quickly to that, if anybody is interested or thinking about stem cell therapy and doesn't know how to kind of navigate understanding what they're looking at, Kerry and I prepared some information. It's not to help you make a decision, it's to help you understand how to read the advice or the marketing material that companies will put out, and for you to judge for yourself whether you think that it's something you want to go forward with. We're not here to make a decision for you, but we can help you understand the jargon and at least keep yourself safe, because I think that's the most important thing. If you're trying something experimental, that you should be very confident that what you're taking is not going to harm you further and that it's going to be safe for you.
Yeah, unfortunately, there's a lot of things out there which will take your money, and that's about all.
Sorry, so if we wanted to get gene tested, we just need to do the two of you?
Yeah, over here.
I'm not entirely sure if it's gene connected, but...
You don't have to find out the result. You can just contribute to the research by having your genes tested. And if you want to find out, you can, but it's optional. You can contribute to the research, but not have to have the burden of knowing necessarily, but a lot of people do want to know anyway.
And as Chris said, if we don't find a gene that's one of those strong genes, then we can't come back to you with kind of a result. But that is a result, because it says that you might not be at the same amount of risk of passing that on as someone who had one of those strong genes.
Thank you.
Just before we come to the next question, and Kerry's got to go.
I'm sorry.
Yeah.
Sorry.
So just like everyone put their hands together. Thank you very much. Safe trip back. Everyone gets stuck in traffic. Thanks very much.
Yeah, look, I'd just like to make a comment. A friend of mine was diagnosed with breast multiple sclerosis, and he was absolutely cleaned alive by people coming up with alternatives. And it caused a lot of stress. He went through a lot of things, and it was really something you really got to consider, because quite frankly, there are people out there who I call snake sale oil people, who will milk you for every dino you've got, will give you every promise that your family wants because you care and love for this person. And please just be very, very careful of that. I'd say to you, stick with MND. It may not give you the time that you love, results that you want, but they're doing the best that anybody can do for you. And a comment for Claire, she is. Yeah, right. She is amazing.
She is amazing.
She's a fighter. She's a battler. The reason why I was a battler. No, she's brilliant. That we said.
Thank you. Any more questions? Okay, if that's the end of the questions, we'll wrap up this afternoon. I just want to again a huge, huge thank you to Claire Riley for launching our research strategy, which we're very, very excited about. Thanks to Dr. Emma Scotter for your presentation, I say, make things very, very simple, and for Professor Chris Shaw for joining us, and we hope, looking forward to seeing you again in December.
Yeah, December.
So a big, huge round of applause, please, for our